Hey everyone, and welcome to another edition of Clark Planetarium's Science Virtual. Thank you for joining us for another cool science activity. I'm Maddie, and I will be narrating. And this is Cynthia, and she will show you everything you need to know to create your very own Extremophile DNA bracelet. But first, let's talk about extremophiles and deoxyribonucleic acid, or DNA for short. An extremophile is an organism that does best under extreme environmental conditions. The suffix phile means love, and these critters love it extreme, like living in boiling water or in acid or other extreme environments that would be absolutely impossible for humans to survive in. For example, did you know there are bacteria in Yellowstone National Park that feel right at home in boiling water? And there's some bacteria that live in your stomach acid. Some extremophiles call deep sea thermal vents home. And the pressure at these deep sea vents can be so great it crushes metal like cans, like soda cans, just crushes it. And did you know there are some microscopic organisms called tardigrades that can survive in space where there is no oxygen and wicked high levels of radiation? Even right here, close to Salt Lake City, we have extremophiles. Have you ever been to the Great Salt Lake? The brine shrimp that live there are considered to be extremophiles because the water is so salty. Each year, over 2 million tons of salt flow into the Great Salt Lake. But because it's a terminal lake, meaning it doesn't drain into the ocean, it's kind of like the Hotel California. The salt can check in, but it can never leave. There is so much salt in the Great Salt Lake that it's 7 to 10 times saltier than the ocean. Now that's salty. All of these extremophiles are made up of deoxyribonucleic acid, or DNA for short. DNA is double-stranded, meaning it has 2 strands of DNA that make it up, forming a double helix. So it's twisted, kind of like a beautiful spiral staircase. There are 4 nucleotides, often referred to as base pairs. Here in this image, we've even marked different colors to indicate different base pairs. They're held together by hydrogen bonds. These nucleotides or base pairs look like this are held together by sugar phosphate backbone. The ability to pass down DNA from parent to offspring and the ability to copy DNA is the key to making and continuing extremophile life, and all life for that matter. The double helix structure of DNA is perfect for replication because the strands can be separated by breaking the hydrogen bonds. And then a new copy of each strand can be made by adding one singular nucleotide at a time, which is exactly what you will do when you're building your very own extremophile bracelet. You will add one nucleotide at a time. DNA is a set of directions. They carry the instructions for life. And this set of directions is made up of only 4 molecules, the nucleotides. And we can think of these nucleotides as letters that make up an alphabet. So the DNA alphabet is made up of only 4 letters. Just like in our alphabet, the order of these letters is important. For example, I need 12 letters to spell extremophile, but in order for me to spell the word correctly, I have to put these letters in a specific order. Otherwise, it just doesn't make any sense. What does that even say? Who knows? Big guess. So, in our DNA alphabet, the order of these 4 nucleotides is what is important. Believe it or not, the 4 nucleotides that make up extremophiles are the same 4 nucleotides that make up this apple. Walk outside, and all the living things you see, trees, worms, cats, flowers, all of it is made up of DNA. Even you and I and Cynthia and everyone here is made up of DNA. Again, it's the order of the nucleotides that make the difference between you, an apple, and an extremophile. So let's talk about these nucleotides so we can start building our very own extremophile DNA bracelet. The 4 nucleotides that make up DNA. The very first one we're going to introduce to you here is called adenine. We're going to use the color orange to represent it and this giant A. So if you have an orange pony bead, it's adenine. The next one we're going to show you is called thymine. The color we've assigned thymine is this beautiful yellow, and it's often denoted as a giant T. After that, we have cytosine. Now cytosine, we're going to make beautiful blue, and we're going to denote it by this big old letter C. There we go, letter C. The final letter we have, or the final base, is guanine. And we're using green to represent it, and this big letter G. Remember that the nucleotides in our DNA are held together by a sugar phosphate backbone, and we're going to use these pipe cleaners to represent the sugar phosphate backbone in our bracelet. The backbone doesn't really store any of the information, but it's important because it holds the nucleotides together and remembers that the nucleotides are in the correct order for our directions. Now, I remember I said the order of nucleotides is important, so the order of how I place the nucleotides along the backbone or my pipe cleaner here is how we will write our DNA code for what will be created. And we will build one strand of DNA at a time. In your packet, you have the directions for one strand of DNA. So let's string that first. I have 2 pipe cleaners to form my sugar phosphate backbone, and each has a different colored bead on it already. The nucleotide sequence I was given starts with a C, so that's cytosine. We're going to put a blue pony bead right on that just to show you all. Okay, so the code in my packet is for this strand of DNA. Keep in mind that yours could be different because you might have a different extremophile than me. That's right, we threw in curveballs. There's multiple extremophiles. You have to complete this activity to figure out what you are. Okay, so I start with this strand of DNA that starts with cytosine. Once I string this nucleotide sequence, I will need to figure out the other side to figure out the extremophile I am, but let's just start by stringing this one. After cytosine, I have thymine, indicated by a yellow bead. Oh, jumpy beads today. There we go. Then I have another thymine, which is another yellow bead. Just keep stringing them on there. Don't push too hard, they'll all fall off the bottom. Then I have 2 adenine in a row. That's going to be 2 orange beads in a row. We're going to keep building this until we've represented all the nucleotides in the sequence that we were given. Okay, so now fast forward, we have our first strand of DNA, and it is made up of 2 strands, so it's time to attach that second strand, everyone. So we're going to go ahead and line up our strings there and twist them together, just so now we have a bottom and it's impossible to push any of the beads off. And we're going to string the other set of nucleotides along our other pipe cleaner. So then we'll finally have our double helix. Okay guys, I'm going to level with you. This is where it gets a little bit complicated. You need to figure out the nucleotide sequence that will go on the other strand. But we didn't give that to you. So how are you going to do it? Remember how hydrogen bonds hold the individual nucleotides together? Well, because of how these hydrogen bonds form, we can only join adenine and thymine together because they're the only ones that will hydrogen bond with each other. So these 2 are the pair in base pair. On the opposite side, we have cytosine and guanine. And now these 2 are the only ones that work with each other. And it doesn't matter if cytosine's before guanine or after, they just like to be with each other. And it can't go with any of the other base pairs. It's always adenine and thymine / thymine and adenine. and then cytosine and guanine, or the other way around. So, let's go back to our bracelet. It's time for us to start stringing in the opposite strand. So, we have a thymine bead, looks like it's next on our parent strand, so we're going to grab an orange adenine bead to complement it properly. And it looks like we have 2 thymines in a row, so we're going to go ahead and grab a second orange bead to represent our adenine. And now, flip the script, we got 2 adenines. So now we're going to throw on 2 yellow pony beads to be 2 thymines. So look at this, we're building a pattern of DNA. We have another yellow, that means we throw on another orange. Uh-oh, curveball, we got a blue. That's cytosine. What goes with cytosine? You guessed it, guanine. And now a guanine? Well, what can go with that besides cytosine? So you build these 2 strands to complement each other, and look at that beautiful pattern. That is your extremophilic DNA. And as you keep going, you'll create a bracelet that looks like this. This is the code. This is the DNA. This is your extremophile DNA, and you're going to make it a bracelet. You're going to put it on your wrist, and it's going to be gorgeous. Look at that. Absolutely beautiful. Voila! That is your bracelet. Now, here's the big question. What extremophile are you? To figure this out, you're going to need to match up that second strand of DNA to the key in your packet. You guys are never going to guess what extremophile I am. Now, the DNA that you've strung on your extremophile bracelet is just a small portion of the entire genome that actually makes up an extremophile. A genome is an organism's complete set of DNA, and remember that DNA is made up of these 4 nucleotides: adenine, thymine, guanine, and cytosine. Bacteria have really small genomes compared to larger organisms, but even Helicobacter pylori, the acid-loving bacteria that can live in your stomach, has a genome that's made up of over 1.6 million base pairs. That's so many, we can't even comprehend it. We're confused just thinking about it. That is so many base pairs. Do you think about your genome? You're way bigger than H. pylori, right? You're huge. And it's made up of over 3 billion bases. Holy cow, guys. If we could uncoil all the DNA and line it up from all of your cells, we could stretch it all the way out to Pluto and then bring it all the way back home. That's a lot of DNA. Thank you guys so much for joining us today. I hope you have fun making your extremophilic bracelet, and we will see you next time. Bye, everyone.